Hello students, welcome to EPG Pathshala. I am Dr. Rajesh Gautam from Dr. Harisingh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. Today I will tell you about a module entitled Inbreeding Coefficient and Inbreeding Depression from the paper Human Population Genetics. So first let's see what we are going to study under this module. We will study or understand in this module about inbreeding, then consanguinity, inbreeding coefficient, inbreeding depression, and we will also know about classical examples of inbreeding depression. So, by parental reproduction as its own importance in natural selection. By parental reproduction has its own importance in natural selection and evolution, in which a limited number of mutations which are not too injurious to be carried by the species furnish an almost infinite field of possible variation through which species may work it in this way under natural selection, but at the same time in biparental reproduction there may have breeding or mating between closely related individuals, for example brother and sister mating and father daughter mating. Such kind of mating practices are called inbreeding. In another word, inbreeding is a mating practice among organisms of common ancestry. It is contrary to outbreeding, which is the mating practice of unrelated individuals. The inbreeding has various pros and cons. In genetics, it is an important practice for retention of desirable characteristics or the elimination of undesirable ones. For course of evolution, variation in gene pool is essential. In the same way, for perpetuation and existence of any species, variation in gene pool should be maintained, and to maintain variation in gene pool, inbreeding should be avoided. But at the same time, to maintain homogeneity in offsprings, inbreeding is essential. Sometimes it is the only way to perpetuate any species or group of organisms in the circumstances of limited number of eligible mating population. Three of the most commonly used definitions of inbreeding are: first, pedigree inbreeding. An individual is considered inbred under this definition when the parents share ancestors. The extent of inbreeding is related to the amount of ancestry that is shared by the parents of an inbred individual. Pedigree information is used to determine the inbreeding coefficient, although originally derived as a correlation as given by Wright 1922. The second commonly used definition of inbreeding is called inbreeding as non-random mating. This refers to the degree of relatedness between mates relative to two mates chosen at random from the population. An individual is considered inbred if its parents were more closely related than two randomly chosen individuals. This type of inbreeding is relative to a random mating population of the same size, although it can be determined from pedigree data. This type of inbreeding is typically measured by the deviation of the observed heterozygosity of an individual relative to the heterozygosity expected under random mating. Out of three most commonly used definitions of inbreeding, the third is known as inbreeding because of population subdivision. When populations are subdivided into more or less isolated groups, inbreeding will also occur purely because population size is restricted and genetic drift results. This occurs even if mating is random within subpopulations. This definition was given by Crow and Kimura in 1970. This third definition of inbreeding corresponds to the mean inbreeding coefficient expected in subpopulations under random mating. Inbreeding is considered a problem in humans because inbreeding increases the chance of recessive, deleterious alleles inherited from a common ancestor. Most studies are concerned with close inbreeding, also known as incest, which usually sets a threshold at the level of first cousin mating. Consanguinity. Two individuals are said to be consanguineous if they have at least one ancestor in common. In the breeding of domestic animals, consanguineous mating are frequently made. Occasionally, matings are made between very close relatives, father or sire and daughter, brother and sister, etc. On the basis of the theory of evolution, all individuals of a species are to some extent consanguineous, since all are descended from a remote common ancestor. Consanguinity and level of inbreeding must therefore be defined as applying only to relationship established after some evolutionary time point at which, for convenience, everyone is considered to be unrelated. In practice, for human consanguinity, common ancestors more remote than a great grandfather are rarely considered. In some human societies, more distant consanguinities may have social significance, but from a genetic point of view, the connection between two individuals who have one great grandparent in common, fourth half cousin, is very big. The occurrence of consanguinity in a population depends on various factors, namely population structure, migration, cultural practices, and so on. Generally, very close consanguineous mating are avoided in human population. Practically, the occurrence of in all human societies in a population is considered on various factors, namely the degree of population structure, relationship, migration, at which cultural practices, mating is and considered so incestuous, may differ very close slightly from one society to another. Mating, but in general avoided parent human population, and brother-sister mating in all human societies. In all societies, incest is considered to be taboo. Incestuous union, the degree of maybe relationship in most of the advanced societies, but mating there is considered negligible. Incestuous proportion may differ in 1967 from one society. Adam to another and Neil have but in general such children offspring, and most of them and brother-sister the result are forbidden sister mating in all societies. Father-daughter mating. So further in this context, let's talk about consanguinity. Two individuals are said to be consanguineous if they have at least one ancestor in common. In the inbreeding of domestic animals, consanguineous mating are frequently made. Occasionally, matings are made between very close relatives, that is, father or sire and daughter, brother and sister, etc. On the basis of the theory of evolution, all individuals of a species are to some extent consanguineous, since all are descended from remote common ancestor. Consanguinity and level of inbreeding must therefore be defined as applying only to relationship established after some evolutionary time point at which, for convenience, everyone is considered to be unrelated. In practice, for human consanguinity, common ancestor more remote than a great grandfather are rarely considered. In some human societies, more distant consanguinities may have social significance, but from a genetic point of view, the connection between two individuals who have one great 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 grandparent in common, means fourth half cousins, is very vague. The progeny of consanguineous parents is by definition inbred. The inbred individuals may carry double dose of a single gene that was present in a single dose in common ancestor. A recessive gene carried in a single dose in a common ancestor may remain hidden until it comes to light for the first time in an inbred descendant. Therefore, recessive traits will occur with increased frequency in progeny of consanguineous mates. An individual who is homozygous for a given gene carries at a certain locus two homologous genes that are physically identical but not necessarily identical by descent. This physical identity is what we call identity by nature. Inbreeding makes an individual homozygous for genes that are identical by descent. Inbreeding coefficient. The measurement of degree of inbreeding can be done by inbreeding coefficient. In 1913, Pearl has given a formula for computation of inbreeding coefficient for each generation. A separate coefficient is obtained for each generation by the formula. The formula is Zn is equal to 100 within bracket 1 minus q n plus 1 over p n plus 1 is equal to 100 within bracket 1 minus q n plus 1 over 2 power n plus 1, where q n plus 1 over 2 power n plus 1 is the ratio of actual to maximum possible ancestors in the n plus first generation. If an individual is inbred, his father and mother are connected in the pedigree by lines of descent from a common ancestor or ancestors. The coefficient of inbreeding is obtained by a summation of coefficients for every line by which the parents are connected, each line tracing back from the father to a common ancestor and thence forward to the mother and passing through no individual more than once. The same ancestor may of course be involved in more than one line. The path coefficient for the path father or sire as to offspring FO is given by this formula. POS is equal to 1 by 2 under root 1 plus Fs over 1 plus FO, where Fs and FO are the coefficients of inbreeding for sire or father and offspring respectively. The coefficient for the path mother or dam to offspring is similar. In the case of grandfather or sire and individual, it can be POG is equal to POS, PSG is equal to 1 by 4 under root within bracket 1 plus FG divided by within bracket 1 plus FO. And for any ancestor A, the coefficient pertaining to a given line of descent POA is equal to within bracket 1 by 2 n under root within bracket 1 plus FA divided by within bracket 1 plus FO, where n is the number of generations between them in this line. The correlation between two individuals, that is RBC, is obtained by a summation of the coefficients for all connecting paths. Thus, RBC is equal to summation PBA PCA is equal to summation within bracket 1 by 2 power n plus n dot into 1 plus BA over under root within bracket 1 plus BB into under root within bracket 1 plus BC, where n and n dash are the number of generations in the paths from A to B and from A to C respectively. The formula for the correlation between uniting gametes, which is also the required coefficient of inbreeding, is FO is equal to 1 by 2 or SD under root within bracket 1 plus Fs into within bracket 1 plus Fd, where RSD is the correlation between father and mother, and Fs and Fd are coefficients of inbreeding of father and mother. Substituting the value of RSD, we obtain FO is equal to summation within bracket 1 by 2 power n plus n dash plus 1 into 1 plus Fa. If the ancestor A is not inbred, the component for the given path is simply where n and n dash are the number of generations from father and mother respectively to the ancestors in question. If the common ancestor is inbred himself, his coefficient of inbreeding must be worked out from his pedigree. This formula gives the departure from the amount of homozygosis under random mating towards complete homozygosis. The percentage of homozygosis, assuming 50% under random mating, is 1 by 2 within bracket 1 plus FO into 100. The inbreeding coefficient represents the probability that an offspring will receive a gene from each parent that is a copy of single shared ancestral gene. The inbreeding coefficient is 0 if the parents do not share common ancestor, and if the inbreeding coefficient is 1, then the offspring has 100% chance of receiving 2 copies of the ancestral gene. However, the maximum inbreeding coefficient of 1 cannot be achieved in human populations. Alternative methods of calculating the coefficient of inbreeding. Falconer 1989 had provided an alternative method of computing F, which is as follows. Ft is equal to 0.25 within bracket 1 plus 2f t minus 1 plus f t minus 2, where ft is the coefficient of present generation, ft minus 1 is the coefficient of the previous generation, and f t minus 2 is the coefficient of the generation before that. It is important to note that recurrence equations can only be used for regular system of inbreeding, that is 3 generations of full ship mating. So, dear students, in context of understanding inbreeding and inbreeding coefficient, we should also know about inbreeding depression. So, let's understand what is inbreeding depression. Biologically, inbreeding has many harmful effects on the offspring. They may be homozygous for certain lethal genes. In this way, reduced biological fitness in a given population as a result of inbreeding is known as inbreeding depression. Many studies have demonstrated reduced survival and fecundity of inbred young. Biological fitness refers to ability to survive and reproduce. Inbreeding depression is often the result of a population bottleneck. A population bottleneck is a sharp reduction in the size of a population due to environmental events such as earthquake, flood, fires, diseases, drought, human genocide, etc. Such events can reduce the variation in the gene pool of a population. After an incident, a smaller population with a few individuals with a correspondingly smaller genetic diversity remain to pass on genes to further generations of offspring. Such reduction population size results in the loss of genetic variation. The robustness of the population is reduced, the ability of population to survive is also reduced. In other words, the higher the genetic variation or gene pool within a breeding population, the less likely it is to suffer from inbreeding depression. Mechanism responsible for inbreeding depression is fitness advantage of heterozygotes, which is known as over-dominance. This can lead to reduced fitness of a population with many homozygous genotypes, even if they are not deleterious. Here, even the dominant alleles result in reduced fitness if present homozygously. For further elucidation of facts to understand the effect of inbreeding depression, some characteristics of man, pig, cattle, sheep, chicken were studied by some of the investigators are given in the table as follows: the name of animals, their characteristics, inbreeding depression, and the reference from where these were taken are given in this table. As you can visualize, man and his characteristics, that is stature or height at age 10, there is inbreeding depression of 2 centimeter. Similarly, the intelligence quotient of men have 4.4% inbreeding depression. In case of pig, the body weight 2.6 kg of inbreeding depression was reported by Faulkner 1989. Then little size of pig was reported as inbreeding depression of 0.24 piglets. Among cattle, the inbreeding depression was estimated as annual milk yield 135 kg. Among sheep, body weight loss was found 0.29 kilogram as due to inbreeding depression. Similarly, in chicken, hatchability was found 4.36% and annual egg production was decreased 9.26 eggs. So, dear students, in this module, what I told you, now I will summarize it. As I told you, marriage between closely related individuals is called inbreeding or consanguinity. So, the consanguineous marriages are widely prevalent in human population. There are several reasons that a population would practice inbreeding that span from religion to geography to royal bloodliness. Inbreeding is a very touchy and controversial subject when it concerns humans, and there is still a lot that we do not know about the possible effects of inbreeding. By inbreeding coefficient, it can be measured. Similarly, the inbreeding depression can be found out. Still, it is very difficult to run experiment to determine all the possible effects of inbreeding in humans because there are too many variables to control. However, most experts would agree that practicing outbreeding will provide a population with the best opportunity to achieve a high level of health. Thank you very much.